All right, today we are going on a pretty wild journey, deep inside your own cells. We're going to see the microscopic assembly line that builds the very stuff you're made of, proteins. Let's dive in.
Have you ever really thought about it? From the muscles that help you walk to the enzymes that digest your lunch, it all comes down to proteins. But how? How does your body know what to build, and how in the world does it get all those finished products exactly where they need to go? Well, the answer is this incredible, massive operation that's happening with unbelievable precision.
So here's the best way to picture it. Think of every single one of your cells as its own little bustling factory. And for this tour, we're going to zoom in on one really important part of that factory, the protein production and export department.
Okay, so before we watch the assembly line go, let's get a look at the machinery and meet the key players that make it all happen.
This right here, this is basically our factory map for the day. We're going to follow the journey of just one protein, one that's made to be exported out of the cell. The path starts at a place called the endoplasmic reticulum, then it travels through the Golgi apparatus, and from there it gets shipped out. Pretty cool, huh?
So you might be wondering, how do scientists even see any of this stuff? This factory is way too small for the naked eye. Well, they use special stains, and you can almost think of them like putting on colored goggles. One dye, a basic one called hematoxylin, stains acidic parts of the cell purple. We call these parts basophilic because, well, they love the base dye. And then another dye, eosin, does the exact opposite, staining the basic parts pink.
Okay, now take a look at this. On the left, you've got a cell that's been stained. See all that deep purple color? To a scientist, that's a huge clue. It means the cytoplasm is just full of acidic material. So what is that material? Well, if we zoom way in with an electron microscope, like on the right, you can see it's absolutely jam-packed with these tiny little dots. Those are called ribosomes. And these ribosomes, these are the actual workers on our factory floor. They are tiny little machines made of RNA and protein, and they have one single job: build proteins. That's it. So now you know, when you see that deep purple stain, you're looking at a cell that is an absolute powerhouse of protein production.
But here's the thing, not all proteins are going to the same place. So it makes sense that our factory has two different production lines. You've got some ribosomes that just float around freely in the cell's main fluid, the cytoplasm. And then you have others that are actually attached to this big membrane system called the endoplasmic reticulum. And believe it or not, where that ribosome is located completely determines the protein's final destination.
So the free ribosomes, think of them as handling the in-house jobs. They make proteins that are going to be used right there, inside that very same cell. A great example is hemoglobin, right? The stuff that carries oxygen inside your red blood cells, or the proteins that make your muscle cells contract. All of those are made by free ribosomes for local use.
Attached ribosomes, though, they're the ones handling the big export orders. These are making the proteins that are destined to be shipped out of the cell to do work somewhere else in the body. We're talking hormones, enzymes, antibodies, you name it. And this export pathway is the one we're going to follow for the rest of our tour. All right, so our journey starts at the first major stop in the export pathway. It's this big network of membranes, and it's studded all over with ribosomes, which gives it a bumpy look, and its name, the rough endoplasmic reticulum, or just the rough ER for short.
So three really important things happen here in the rough ER. First, the protein gets built. That's called synthesis. Second, it gets modified and basically goes through quality control. And third, it gets all packaged up for transport to the next station on the line.
Okay, now this part is amazing. As the attached ribosome builds the protein chain, it literally feeds it or threads it directly through a channel right into the inside of the rough ER. That inside space is called the lumen. And this is so clever because it immediately separates the new protein from everything else in the cytoplasm. It can't get lost, and it can't mess with any of the other factory operations.
But once it's inside, the protein is still just a long, kind of flimsy chain. It's not useful yet. The ER is where the real magic, the quality control, happens. The chain gets folded into its precise three-dimensional shape, and this is absolutely critical. If the shape is wrong, the protein won't work. It also gets a few other chemical tweaks, like adding some sulfur groups or some sugar tags in a process called glycosylation.
So, once the protein is folded just right and it has its proper tags, it's ready for the next step. A piece of the ER membrane actually pinches off, forming this little bubble with the finished protein tucked safely inside. This bubble is called a transport vesicle, and it's basically our little cargo container ready to ship out. So where is this little package headed?
Well, it's on its way to the factory's central post office and shipping department, the Golgi apparatus.
Yeah, if we look back at our factory map, you can see those little transport vesicles budding off the ER and making their way over to the Golgi. They're about to hit the next critical phase of the whole operation, processing and sorting.
You know, the Golgi itself kind of looks like a stack of flattened pancakes. It's got a receiving side, called the sysface, that's the inbox. Our little transport vesicle docks there and merges with it. The protein then travels through the stack of pancakes, getting a few more modifications along the way, until it finally reaches the shipping side, the transface.
So the Golgi's main job is to put the finishing touches on our protein and get it ready to be shipped out. It does some final chemical modifications, but maybe most importantly, it acts as the master sorter. It literally reads those sugar tags that were added back in the ER and then packages the proteins into brand new vesicles based on where they're supposed to end up.
And here we are, the final step in our protein's journey. A brand new vesicle, what we call a secretory vesicle, packed with our finished protein, buds right off the transface of the Golgi. It travels to the very edge of the cell membrane, it fuses with it, and then poof, it releases its precious cargo outside the cell. Mission accomplished. Okay, so we've followed one tiny protein on its incredible journey. But let's pull back a bit. Let's zoom out. Why does this microscopic assembly line matter so much?
Well, the answer is pretty much everything. This exact process is happening constantly in countless cells all the time. The insulin that keeps your blood sugar in check, the digestive enzymes in your stomach, the antibodies that fight off infections, every single one of them is made on this exact same factory assembly line.
When you really stop and think about it, it's just mind-boggling. Trillions of these microscopic factories are running inside you right now, 24/7, with this breathtaking precision, all just to keep you going.
And that really brings us to a final crucial point. This whole intricate process isn't just cool biology, it's fundamental to life. A lot of diseases are caused by just one tiny error somewhere in this factory. Think about cystic fibrosis. That's caused when a single protein folds incorrectly way back in the ER. Quality control catches it and destroys it, so it never even makes it to the cell surface where it's desperately needed. It really makes you wonder, right? What happens when just one step in this incredible assembly line breaks?